Good afternoon, everyone. My name is Chunhong Mao. I'm a member of CIA Division, and today I'm going to talk about one of the projects that I've been involved, and it's GPA Project. So GPA Project is funded by NIH, and it's a large collaborative project. And the goal of this project is to identify genetic and proteomic characteristics of early onset atherosclerosis disease. And 6 institutions are involved in this project. Wake Forest, Dr. David Harrington is the leader PI, and they're expert in cardiovascular genetics analysis. And Louisiana University provide the clinical sample for us. The UT Houston provide genomic sequencing. JHU and Cedars-Sinai, they provide proteomics data. And for VBI group, sorry, the slide is kind of low. We basically provide the bioinformatics support for the whole project. And we also provide data management and project management. The member of the group from BBI are Becky Will, Dan Sullivan, myself, and Andrew. And we also have people from Northern Virginia. They're Dr. Joseph Wang and Dr. Guoqiang Yu. So today I'm going to just focusing on talking about the workflow that we developed for the project to identify protein genes that are important for the disease. And we combining both the proteomics data and GWAS data for the analysis. And also we provide the detailed functional analysis for the project. So this is a simple graph presentation of the workflow that we developed. Starting, you see, trying to get the, okay. Let me go back to the slide, okay. Okay, starting from the proteomics data that are provided from this project, we first identify the significant differentially expressed genes that are important comparing disease cases versus non-disease case. And also from previous GWAS data, the genome-wide association study, we have a list of genes that are known to be associated with vascular diseases. And from that list, we can build a network model to identify genes that are important in the protein-protein interaction network. And by comparing the two lists, we can look at what are the genes that are similar between these two studies and what are the differences between the two studies. And we then go on to look at more detailed functional analysis of these gene candidates. So the detailed analysis include a gene ontology enrichment analysis, the protein-protein chemical and protein-chemical interactions, the pathway analysis, and the associated disease and functions. And then we can look at whether there are genes that are good for drug targets and any of the toxicity information. So here is one example of gene ontology analysis result. Basically, gene ontology provide controlled vocabulary terms that describes biological processes, cellular components, and molecular function for gene products. So as this result, we can immediately see some of the gene ontology terms such as very low density lipoprotein particle remodeling, the lipid metabolism, that are important terms or process or pathways that are possibly involved for atherosclerosis. And we can also visualize these terms and to look at the relationship between them. So, and also it's important to look at how these proteins interact with other proteins in the system, and look at also these proteins, what chemicals can possibly interact with them. So this is an example of a protein-protein network and protein-chemical interaction network. And we can see, interestingly, that two chemicals, cholesterol and triglycerol, that are important, we know, to be involved in the disease processing. So another detailed functional analysis we do is to look at what pathways that these proteins are possibly involved in, and what disease and functions that they're associated with, and what are possible drug targets based on the known knowledge base, and then what are the upstream like regulators and the regulatory effect networks. And then we can also look at the metabolite profiles for these proteins. So based on existing knowledge, the annotation of these proteins and pathways, we can actually build a cell model to look at the where these proteins located in a cell, whether they're intracellular or extracellular, and what other proteins that they are interacting in the cell, and how they signal to each other. And also we can map the pathways on top of these genes to look at the relationship. And this way we can, it will help us to generate hypotheses. So for the future work, we're going to continue to refine our workflow in terms of finding good software and databases and adjust parameters. And also, we're going to continue to do functional analysis for any new data that are generated for the project. And then we're going to integrate the genomic and the proteomics data and analysis result. Hopefully, it will generate some interesting result. Thank you.